From MicrobeTV, this is Office Hours for Wednesday, January 7th, 2026. Happy New Year, everyone. Yeah, we're in 2026, and who said it couldn't be any worse than 2025? It's already worse. Oh my gosh. I'm Vincent Racaniello, and welcome to your viral corner of the internet where you can vent and you can ask questions about viruses. You can learn things from me and from each other there in the chat. Welcome, moderators, tonight. We have Vanity Nutrition. We have Tom Steinberg. We have Lef's Fabi. Thank you for moderating. And everyone, tell me where you're from. I'll shout you out in these early minutes. We've got a full agenda for you tonight. We're going to go over this changed vaccine recommendations by RFK and his ilk. We will have a mini lecture. Someone asked me to explain how the poliovirus receptor was identified. So I dug into my old images, and I'm going to do that for you. And finally, of course, some poetry. Tonight is another one of my favorites, E. Cummings, he of the no capital letters and really unusual line breaks and creative spacing and so forth. I'll have to show some of these to you to see how they get aligned. But in the meantime, let's see who's here. And of course, horrible news tonight. ICE kills a woman in Minneapolis in cold blood. The guy just shoots her in her car. She's trying to get away. Who would not want to get away from ICE? They're all assholes with no training and no self-restraint, no empathy whatsoever. Killed this woman who is a U.S. citizen with a 6-year-old daughter. And of course, the administration is lying. They said she was trying to run the guy over. If you look at the videos, it's clear she was not. Stop friggin' lying. I'm so tired of it. Even RFK, all the way down to these lackeys. Oh, my God. But we're going to do virology and science tonight. Let's see who's here tonight. Rita. Where did it go? Oh, Rita, hi from the sagebrush steppes and canyons of Southwest Ohio. 42F, 5.5C sprinkling. We're experiencing a disturbingly warm winter. Well, here it's been frigid, below freezing every day, except today, for the first time, it's 5 degrees Celsius. It's been above freezing since this morning. So that's cool. I actually can feel it. I don't need to wear a cap. Oh, my gosh. Spamly, hello, happy New Year. Kathleen is at the Jersey Shore on a warm January. Well, I don't know about 5C being warm, but it's warmer, that's for sure. Tom is in the rain-soaked Oregon Coast Range. Welcome. Will is in China, 5 degrees and sunny. Changsha. Kang is in Chicago, 4 degrees C and clear. Vanity Nutrition is here. Welcome. Yeah, Visto is in Australia. I saw the ICE gangs killed that poor woman in the street. The USA looks so scary. No law and order. You got to be careful. You got to be careful. They're disgusting. They're worse than disgusting. You know, they get paid a lot and they're not trained to have restraint. You know, if you think someone's driving at you and she wasn't, you shoot their tires out. You don't have to kill them point blank. Morons. I hope something comes of this, for God's sakes. Hello, Visto. It has cooled from yesterday's 41C. Whoa, that's hot. Oh, my gosh. Brian is in Knoxville, 55F and humid. Do you do tours of the incubator? Of course. Just, you can send me an email, vincent@microbe.tv, and just tell me when do you want to come, because I have a schedule. And I'm usually there, but not always. Sometimes I have class or other things, or a podcast or 2 or 3. But send me an email and we'll work out a time. Yeah, I've had a bunch of people. And now we have a guest book that gets signed. Yep. Carol, our favorite nephrologist, is in Bakersfield, where it's 12 degrees Celsius. Leo, hello everyone. Greetings from the Mendocino coast, where it's 9.44 C and damp and wet. Not able to make it here for the last 3 weeks, and it's wonderful to be back to the nest. Yes, the nest. It's good. It's not a thermostat. It's a place. Hello, John from Minneapolis. 37-year-old widow leaves behind a 6-year-old. I will call what happened a homicide. It is a homicide, and this guy better go to jail for it. It's just disgusting. And they should get the hell out of Minneapolis. They should get the hell out of all cities. This whole business is reprehensible, abhorrent. I don't know anyone who wants this. I don't know anyone, but I'm sure there are people out there. Black and yellow. That's right. Although I'm just wearing black with a red splash tonight. But yes, if you want to know how to pronounce my name, it's Black and Yellow. Vanity saw in the news a small child died from flu-related transverse myelitis. Yes, it can happen. A lot of flyers were masked yesterday on my flight between JFK and Salt Lake City. I always mask on flights, on trains, on subways, for sure. Hat is in the San Fernando Valley, 62F. Hello up to code. Welcome. Brian is in Montreal, minus 2C. Still freezing up there. Pete is in London, where we have winter hovered up and down from freezing yesterday, lurking at 3C tonight. Elizabeth, I'm so screaming angry about ICE in Venezuela. How about the vaccine changes, too? Scream about that, too. I can't even talk. Hi from West Virginia. And John says it's 1C in Minneapolis. Kent is in Bellington, Washington. What's worse than RFK? What's worse, RFK or Greenland? The Venezuela, Greenland, Cuba, Mexico, wanting to annex all these countries. It's disgusting. It's not what we do. Well, we used to do it, unfortunately. Big pieces of the U.S. came from other countries. But we bought them, right? I don't know. Greenland hopefully wouldn't kill anyone, but RFK is going to kill people by messing around with vaccines, for sure. We'll talk about that for sure. Soul Symphony, welcome. Kent, thank you for your support of science communication. We really appreciate it. Up to code is in Nova Scotia. It is a dark and stormy night. Do I know Lee Hood and the folks at the Institute for Systems Biology? I don't know him personally, but of course I know of his work. Very famous. Mike is in Burien, Washington, 4C and sprinkling rain. Privyet Veronica. I'm starting to forget your name, right? I know you like Jellicle Kitten. Moscow, -6, lots of snow. Sandra's in Mexico, 12 degrees Celsius. Science, I love it. Yes, we're here for the science, but I have to let you know of my outrage because you're all outraged too. Carol got my second Shingrix ipsilateral arm. That's the same arm. And pneumococcal vaccines I'm good for this year. So the Shingrix is going to protect you against dementias. And we're doing a paper Friday on TWIV, which, the original was one kind of dementia. Now they look at all different kinds of dementias, and because there are multiple kinds, and it protects against many of them. So it's very cool. Pete says, I'll eat my hat if big oil invests much in an unstable Venezuela during low crude oil prices. Yeah, there are a lot of reasons why they're not. It's the infrastructure is old and requires a lot of money. The oil is this heavy stuff that's not easy to refine. It's just not thought out at all. He's an idiot with no information behind any of his decisions. Truly an idiot. Viruses and senescence. I don't know much about that, but I could learn. So I have here Mobakia. I wrote it on my sticky. Now we did that last week. Okay, so I'm going to write down Viruses and senescence. So you want to know what role viruses have to do in senescence, right? You know, aging is a real it's a complex thing. It's mainly a metabolic disease. You know, your body starts to fail at doing respiration, right? You know what respiration is. It happens in the mitochondria. Electrons get added to oxygen, the terminal oxygen acceptor. You get water and you make energy. Very efficient ATP generation there in the mitochondrial membrane. As you get older, the process fails. Read, what's the book by Nick Lane that I just recommended on Twiv? Transformer. Read it. It'll tell you all about metabolism, cancer, aging. It's amazing. I have to go back and read it again. And I think I need to get a hard copy so I can underline stuff and take notes, because I can't do that I can underline on the Kindle, but yeah. Les says, children have died from flu, and none of the news stories point out how people can protect their children. Of course not. I mean, the news outlets could do that. A lot of them are pointing out that the move by RFK is really bad, fortunately. Lori is back in Texas after spending the holidays in Canada. Minus 12 C to 25 C. I'm missing the snow already. Soul Symphony 4 C in Minnesota. Better than minus 12 last week. Legotic, Chicago, 4 degrees Celsius. Maureen is in Northeast Ohio, 41 F. MN warming up this week in D.C. Jane is in Michigan, 36 F. Rona's out on Long Island, a very sad day today. Yep. Brutus is in Santa Barbara, 14 C. Kelsey, hello from, okay, hello. There you go, that name, nice and warm, 21 C in coastal India. Hello, coastal India. Truptisatish Kadni. Okay, welcome from India. You're far away. Yeah, news has been pretty abysmal. I'm praying for boring news. I don't think you're going to have boring news until the elections. Nope. DOJ admitted they've only released 0.6% of the Epstein files. 2 million more are being withheld, and the ones released are all blacked out. It's just ridiculous. Liars. Perpetual student. Hello from the D.C. metro area. The temperature is 9C. My coworker and I are watching from work. We currently have Staph aureus vaccine in clinical trial. Very good. Very good. When you get your results, we will talk about it on TWIM. That would be awesome. Welcome. Kelsey, yes. Hello from SoCal, reeling from today's news. Rest in power. Yeah. Brian is in Sydney, where it's bloody hot. Michelle is in Philly. Back from Italy, 44F6C. John is in Thailand. Beautiful but windy, 22C. Buddha is in Illinois, it's 9C. Abdul Rahman is in Brooklyn. Welcome. Good to see you again. So, speaking of visiting, Abdul came by not too long ago, last year, to visit the incubator. So, yeah, you can come by, for sure. Outrage is the only sane reaction, that and sorrow. Keep fighting the good fight. Yeah, I mean, I'm not a politician, but I react to political news and horrified news, and I have, my strength is science, and we can tell you all about that. But I have to share with you the pain of all this other crap that's going on. It's ridiculous. I mean, I don't want Venezuela. Leave him alone. The guy is a crook. You can't go and take a crook and try. I mean, what did he do to you? He's a crook in his own country. It's ridiculous. Yuxing, what is it? 7F? Can't be. Hello from Hangzhou. Sunny winter morning, 5C. Oh, that's part of your name. I'm sorry. I thought the C and the F were Celsius and Fahrenheit. So it's 5C. Welcome. Is in Thailand, 22, our second tie. That's pretty cool. Kang, headline from the Chicago Sun-Times. Illinois won't drop the number of vaccines for children despite the new federal guidance. Many states are not going to go along with it because it's pure madness. There's no science behind it whatsoever. I'll talk about that in a bit. Oh my gosh. Soul Transformer, yeah. From an evolutionary perspective, what would a persistent virus benefit from inducing senescence? Like HIV and its like. Wouldn't matter if it's already found another host, right? That's the prime goal, is to find a new host. And so if senescence is a long-term effect, then it's probably already spread. But yeah, that's the other thing that Nick Lane talks about in the book, The Evolution of Metabolism. It's really good. How safe are from Buddha? How safe are vaccines compared to all other pharmaceuticals? Are they our best medicine? Well, it's about prevention, right? You get a shot or two, and you're protected from severe disease. And the side effects are generally mild to moderate. You don't die for the most part. So I would say that's the best bang for your buck. You know, if you don't get vaccinated and decide to do antivirals, then every time you get sick, you have to take it, assuming there is one for your virus, right? So we have them for SARS-CoV-2. We have them for influenza. We don't have them for RSV. Have them for hepatitis B. We don't have it for rotavirus. We don't have it for polio. So sometimes you don't have a choice. Not a lot of antivirals against acute infections. So for HIV, that's all we have is antivirals. No vaccines. The antivirals you have to take every day the rest of your life, except lenacapavir, which is now twice a year, which is brilliant. But we did talk about that. But they have side effects. The HIV drugs have side effects, for sure. Anyway, they don't kill you, but they have side effects. They make your life kind of miserable. Lori finished Breathless by David Quammen. It's recommended. Yeah, Breathless is very good. I have it somewhere here. I've read most of his books. Yep. It's a good title, right? Vanity. I just saw Governor Walz has prepped Minnesota's National Guard to remove ICE agents. That's going to be interesting. I don't know how that's going to work. But you should get them out. Get them all out. Just wrap them up and throw them out, wherever they came from. Hello, David from Dayton, 3C. Yeah, for whom the bell tolls. Ask not for whom the bell tolls, because it tolls for thee, or something like that. Let's get it right. Let's look up the right. Oh, God. For whom... Yeah, by John Donne, right? Donne. Oh, we can read it. John Donne. For whom the bell tolls. No man is an island entire of itself. Each is a piece of the continent, a part of the main. If a clod be washed away by the sea, Europe is the less. As well as if a promontory were, as well as if a manor of thine own, of thine friends were. Each man's death diminishes me, for I am involved in mankind. Therefore, send not to know for whom the bell tolls, it tolls for thee. Each man's death diminishes me. Frickin' Murphy, is that his name? The mRNA guy who went to the dark side. He said 250 pediatric deaths from flu is not much. It's minimal. Every death diminishes you. It's somebody's kid. Robert Malone. What an a-hole. I'm sorry. I hate to call people names, but these people are deserving it more than anybody else. Oh my gosh. For whom the bells tolled. Yeah, the tangled tree is also good. The administration is heartless and a bunch of bullies and liars and ignorant and malignantly, what is it? What did I call it? Malignantly ignorant, yes. Kelsey writes, I'm sure you'd love for politics to be irrelevant to your work here, but it's not. No, I know, but what ICE does is not relevant directly, but it still infuriates me. And I want to share my fury with you guys because I know for the most part, you're infuriated as well. Veronica says, some sane person said it's no big deal that meningitis vaccine is off the schedule. Oregano has antibiotic qualities. Oh, yeah. Great. JM is in Germany, -1 and snowing. Love what you do for science, Vincent. Thank you very much. I have a big team doing science with me. I'm very proud of it. And like this tonight, I love that everyone's here and we're going to talk. A milligram, Kang says, of prevention is worth a kilogram of cure. Nevitz in New York City this evening, 7C, top of Mount Washington, -8C. Spillover by Kwame is also very good. And, of course, Kwame has been on TWIV twice, I believe. Patricia writes, CDC was effectively doctors or providers' word, took so much weight off the doctor because all the nurse had to do, depending on the state, was look at the vaccine schedule and look at the patient's history and order up the vaccine. Yep. Malignant incompetence. Yes, thank you. C. King from BC, 3 degrees Celsius. Hello, Tryon from Romania, right? Maybe you're not there now, but you're from. Did they use intradermal inoculation to test mRNA vaccines? No, I don't believe so. They did intramuscular. That's how it was done in people, right? One death is too many. Absolutely. Totally agree. I mean, they're just saying this crap to try and make a point, but that's Malone said this at a testimony committee, and that's just, in my opinion, the worst thing you can say. 250 kids' death is not much. Those are 250 kids, people's kids. How can you say that? It's so cold-hearted. Buddha writes, reducing childhood poverty in the U.S. by 50% would require an estimated annual expenditure between 70 and 112 billion. We're paying 170 billion for ICE to kill citizens. This is just ridiculous. This country is so screwed up because it's geared to billionaires, to rich people. Okay, 70 to 112 billion is nothing to cut poverty by 50%. We also should have a healthcare system. It's fragmented and crap and too expensive. So many people fall under it. They don't get good healthcare. They get sick, and that's why we have a lot of sick people here. And it's going to get worse with this new vaccine schedule. Nevitz writes, we went from no man is an island, FDR, JFK, to rugged individualism of Reagan and Thatcher, which eventually brought us Drumpf some 45 years later. This is the pinnacle of what the Republicans have been trying to do for years, except these guys are really evil. The Republicans don't want to help people who need help, right? That was what FDR was all about, right? Helping people with Social Security and Medicare and so forth. Now, since Reagan, they've said all these systems are dragging the rich people down, so they can't make as much money as they can. So we're going to get rid of them. And so that's what we're seeing today. And this is going to fail, and the whole thing is going to be a laughingstock. In November, we're going to rip a new opening in these people and flip Congress. We're going to convict Trump and kick him out, and convict RFK and kick him out as well. That's the goal. Get a decent HHS head. Do I wear a mask when shopping? It depends how crowded it is, right? The grocery stores are very well ventilated. There's a lot of air movement. If it's really crowded, yeah, I'll wear a mask. But when I go on a Sunday morning and there's nobody around. Yep. Pete writes, next one kicked over from science denial is the National Center for Atmospheric Research. Right. Destruction of knowledge and expertise. Pure spite. It damages the whole world, not the USA. Yep. Have you heard about a fentanyl vaccine? I have not. Give me a break. Just stop taking it. Let's see. Let's look it up. University of Houston. Yeah, they made anti-fentanyl antibodies that prevent it from entering the brain. So now they have to figure out how to induce such antibodies, right? It's not a bad idea, but really, you look, viruses you can't avoid sometimes, right? But you can avoid fentanyl. Oh my God. And we have the childhood vaccine schedule that matches up with a peer country without universal health care that the peer country has. And other things. I mean, Denmark is less than the size of New York City. It's a homogeneous population who do what they're told. And they have universal health care, which pays for everything. But they don't want to pay for every vaccine. And as Paul Offit pointed out in the latest Beyond the Noise, which will come out Monday, Denmark has as many cases of rotavirus per capita now that the U.S. did before the vaccine. We used to have 70,000 cases a year, completely eliminated by the vaccine. Why the hell would you eliminate that vaccine if it's getting rid of 70,000 cases a year? And don't tell me it's not a lot of cases. Oh, my God. So a lot of people are wearing a mask. Helen and MN and Angelical. I take chances because I take chances. Okay, like at the incubator, we had Immune 100. I should have masked. I'm not going to greet people. I'm not going to do a podcast with a mask. I'll take a chance with my health because I think it's worth it. But you guys should all mask. I agree. Nevitz, it's estimated that Trump's malign neglect and incompetence in handling the COVID pandemic led to 300,000 excess deaths. Not a peep by the lamestream media. By any other country, there would be hearings. He should be in jail. January 6th insurrection, for God's sake. He organized it. It's disgusting. Yeah, Mark fears there may never be another free election in this country again. There will be. There will be. I must look, Lagotic says, for the 2 episodes with David Quammen. I remember enjoying his writing back in the day of Soldier of Fortune and Outside magazines. We have both evolved, I hope. Jane writes, I have to go to a funeral viewing tomorrow, and Michigan flu season is in full swing. And Veronica writes, if you all are abducting dictators, I wonder if Orange Man takes requests. I know what you have in mind, for sure. But another one would take his place. Michael, hello from Mexico City. 72 to 11 childhood vaccine shots is their goal. Are those the accurate numbers? I don't know what the numbers are. I can tell you the number of vaccines. They've gone from... Well, I'm gonna cover it in a minute. Just wait. But, you know, Trump has got his eyes on Mexico, too. It's ridiculous. The rich, Kang says, are ignoring the source of their wealth, according to Adam Smith. The rich make money by having other people work for them who are not rich. Okay? That's the way it works. That's how I learned in college. This is how economics works. There always have to be some downtrodden people to make the billionaires rich. If everyone's sick, if 10% of the population is sick, you're going to have a hard time. Abdur, do you think we can have a rebirth in the country without bloodshed? I do. I think that we're working through the courts, and the Democrats need to get their act together. They need to come up with someone who's appealing, and they have to, like the lady in South Carolina, right, running against Lindsey Graham. She said her motto is, it's me or measles. She said, I never thought I'd be running on that platform. That's what you have to do. right? Point out all the fallacies that are going on. I don't think we need bloodshed, no. Naltrexone is an FDA-approved once-monthly injection used as part of a comprehensive treatment program to prevent relapse in people addicted to opioids, including fentanyl. Right. That's right. We talked a little bit about that on Twin a couple of months ago, and the ads for this are all over the trains. You know, your little inhaler. Carry it with you. Regarding fentanyl, addiction is a tough one to deal with. I understand. Okay, make a vaccine. Here in China's winter, I wear an N95 outside because of high pollution. Veronica says, to take a country back, you need nonstop protests, and those are dangerous. Authorities will do anything to stop them, but I don't think there's any other way. Yeah, you got to get rid of ICE because they're going to just shoot people. What's your opinion about old vaccines such as Bordetella, diphtheria, pathogens that have gone out of news but lurk around? We have a pertussis outbreak here in the U.S. because people aren't getting vaccinated. They're there. These are in the environment. You have to vaccinate against diphtheria and DPT, diphtheria, tetanus, pertussis, for sure. There's no question. This is what happens. You have a pertussis outbreak. Jane writes, my husband and I have funeral viewing tomorrow. I know there will be a lot of people. We are in deep red flu cases. We are both vaccinated. If you were me, would you wear a mask? Yeah, I would. Oh, see, I don't have to worry about podcasting at a funeral. So that's the thing where, yeah, I mean, I'd definitely wear a mask at a viewing. Yeah. To the neocons and Marco Rubio, Cuba is the big prize. If you don't want to take a chance of getting sick, wear a mask. No one seems to mind. I agree. But I pick times when I don't. And like, if I go to a science meeting, I'm not going to wear a mask. I want to talk to people and have them see my face. If I try and stay away from the crowds. But did Dr. Griffin say you can take Tamiflu as a prevention if exposed to someone with flu? I think he basically said that. Yeah. They said, so if you're exposed to someone, yeah, you could take it. And it's an, I don't know, 5-day course. And then if you're exposed again, you could keep taking it. Yeah. Okay, everybody agrees with that. Yeah, 40% of pet owners are vaccine hesitant. It's ridiculous. It's crazy. Visto, Vinny lecture on CRISPR was great. I almost understand. Has there been TWIV on tandem interspace guide RNA? No, there hasn't been. There's going to be some rabies. Yep. Julie said, I just flew back and forth Newark to Denver. I wore KN95. Upon walking into the airport and until arrival and getting outside, N95 just hurt my ears after so many hours of wear. I wear a KN and I put it on once I'm seated. And then I leave it on tour up in the air. Okay, you can make a vaccine for fentanyl. I've been told that it's a problem. It's hard to get over your addiction. Okay, fine. If someone had flu in 1968, are they perhaps immune to this year's flu? Well, they're not immune. They're going to have modified disease. But this H3N2 that's circulating now has drifted quite a bit from that one. But they should have T cells that will protect them against severe disease. Yeah, that's correct. That's what Daniel said. But he said nobody uses Xofluza anymore, just Tamiflu. Hello, Praveen, big fan from the UK. Loved your episode with Luis Villarreal from years ago. Oh my gosh. That was a long time ago. I haven't seen him in ages. He retired, and I don't know what he's up to. Jonathan is in Encinitas. We're at 16C. Welcome. Was the mini lecture on CRISPR on office hours? Yeah, it was part of a gene editing mini lecture. Yeah. Carol wants to know, my son will be flying to New York for work. Should he quarantine upon returning home to protect his 3-month-old baby? He will, of course, be masking. I would just mask. I don't think you have to quarantine. If he shows symptoms, then yes, he should. But I would say just mask. That's what I would do. Tamiflu is a 5-day course for treatment at 1 capsule twice a day. For post-exposure, it's once daily for 10 days. So what do you mean it's a 5-day course for treatment? Treatment implies that you're exposed. I don't know what you mean. When flying, I always open the air vent above my seat. That's right. Have the air coming down. Hello, Juan. How was my birthday? Quiet. Didn't do anything. I worked. It was January 2nd. I did a TWIV and a TWIP, and I was in heaven. Spamlet writes, Cuba is the only country to be declared environmentally sustainable by the World Wildlife Fund for Nature. We should be following its example or even get free health care for all. They're also very good at vaccinating. They eliminated polio before we did. We should leave them alone. Hello, Frank from Houston, 22C. Can someone explain why Homo sapiens have done just fine over the last 260,000 years? No, Homo sapiens hasn't been around for 260,000 years. But anyway, without vaccine, and now within the last 80 years, everyone needs it. Okay, Thomas, you don't know what you're talking about. Homo sapiens did not do fine, did not do just fine. Most people died at 30. Look up your history before you make stupid statements. We developed vaccines, and now the lifespan of humans is over 70, 75 years. You used to die in your 30s, and when you were a hunter-gatherer, even sooner. Don't be silly. Vanity, I thought single-dose Xofluza is one with better data. Yes, Xofluza is a one dose, and it works better. The one that's not used is Relenza. The amantadines are not used anymore, but that's a different target, right? I'm talking about the neuraminidase inhibitors, Relenza and zanamivir, Tamiflu, and Relenza. Relenza is not. Daniel said last week Relenza is not prescribed very much. JM, gotta go. Big fan of you and all the other hosts. Please keep in the fight for truth. We will. Thanks for coming. Yes, lifespan was 30-year-old plus you get sick often. Right. Thomas is just, what do you call, baiting us. Homo sapiens haven't been, you don't get any of your facts right, okay? Homo sapiens hasn't been around for 260,000 years. Okay, it's 8:40. Has Dr. Griffin ever taken you out on his sailboat? No, he never did. I never pushed it. He's always saying, Vince, you have to come on my sailboat. I said, okay. And then nothing happens. It's okay. It's totally okay. You know, people are busy. One day, hope to do it. Treatment is for symptomatic disease with symptom. Prevention is prior to I see, post-exposure. I got it. We use Xofluza thanks to Dr. Griffin and yourself. Actually, it was Bob Krug who wrote in, because Xofluza is an endonuclease inhibitor, and Bob had discovered the endonuclease many years ago. Yeah. Yeah, here's more data from Frankie. I don't know about the 30s, but people mostly died at like 5 years old. Yeah, a lot of kids died. Child mortality was insanely high back in the days, and vaccine was one of the major things that brought it down. In fact, in the early 1900s, the mortality was about 40-ish, 50, and now it's gone up, but it used to be 30-ish. You'd get infectious diseases or you'd be killed by someone who was pissed off at you. Humans did not, in fact, have a good time all those thousands of years. That's why they did everything they could to find out ways to treat illnesses. Google says Homo sapiens have existed for 300,000 years. Maybe Homo has, but not sapiens. Let's look it up. How old is Homo sapiens? Now I'm going to have to eat crow here, right? 300,000, archaic Homo, modern from 160,000. Okay, that's the difference. We're not talking about archaic Homo. I'm talking about modern, which is about 150,000 years. Okay. Hello, IF specialist from the St. John Valley in Maine, where it's warmed up to 27F. Yeah, just go to look in an old cemetery. You'll see a lot of people passed away quite young. Is Tamiflu over the counter? No, you need a prescription for Tamiflu. All right. Got to do a mini lecture. No, I got news for you. No mini lecture is next. Okay. So as you know, RFK has made changes to the vaccine schedule. Okay. He didn't have to. There's no reason to do this. But there are lots of articles about this. You've seen them, so I'll just show you one. But the Times has a good one, but it's paywalled, and I don't have a subscription because I got pissed off at them years ago. Here. And this one is Axios. Trump administration makes deep cuts in child vaccine schedule. So they cut the number of shots to 11 to align it with what it calls consensus peer nations. Now I can't get into the article. You know, it's weird. Trump doesn't want to be like any country, but he says we should be like Denmark. So there's no consistency in this stinking administration. Where did I put the Daily Beast? Yeah, there we go. That's another article. Anyway, there's a very nice chart in the New York Times, which I got. Daniel has it up because we're going to talk about tomorrow, so I'm going to show it to you. But you know, this senator from Louisiana. This is from Obsessed, the Daily Beast. Republican who cast key vote for RFK Jr. rages at his vaccine changes. All right, so this is Bill Cassidy. Okay, William Cassidy. There he is. You know, he was on the subcommittee that approved RFK's nomination. He could have voted no, but he said RFK promised he would not change the vaccine schedule, and he promised he would not change the composition of ACIP. And he said, yeah, I won't. And he did it because he just frickin' lied through his teeth. I mean, Cassidy, come on. What do you think these guys do? They're lawyers. Now he's been left furious after the raw milk enthusiast ripped up the National Pediatric Vaccine Schedule recommended by CDC. It roadmaps routine shots for children and babies, but has been replaced by a new version that no longer recommends immunization against rotavirus, influenza, meningococcal disease, or hepatitis A. And of course, COVID was already removed a while ago. Trained physician Cassidy was unable to hide his fury at RFK Jr.'s new schedule, which cites the loss of public confidence in vaccination for the change. This is BS. That's not data saying you have to make a change. It aligns the CDC's recommendations with Denmark, despite the Scandinavian countries' widely different demographics from the U.S. and also healthcare, universal healthcare. So here's his X tweet where he's supposedly furious. As a doctor who treated patients for decades, my top priority is protecting children and families. Multiple children have died or were hospitalized from measles, and South Carolina continues to face a growing outbreak. 2 children have died in my state, Louisiana, from whooping cough. All of this was preventable with safe and effective vaccines. The vaccine schedule is not a mandate. It's a recommendation giving parents the power, changing the pediatric vaccine schedule based on no scientific input on safety risks and little transparency will cause unnecessary fear for patients and doctors and will make America sicker. Yeah, that's great. Why did you trust him? It's not to be trusted. Unbelievable. All right. So what exactly did he do? So, I mean, this comparison with Denmark is just BS, right? And other countries give fewer vaccinations, right? Would it go from 18 to 11? But every country is different. The population is different. Healthcare is different. Denmark made the decision not to do rotavirus and influenza vaccines for young people. It's a monetary decision. They decided they didn't want to pay for it. So here is the change. This is from the New York Times. And as I said, Daniel has it up. I hope you can see it. So in the red, what's no longer recommended for all children, okay? At birth, Hep B first dose, respiratory syncytial virus first dose, no longer recommended. 1 month, hepatitis B second dose, no longer recommended. Then at 2 months, we get DPT, Hep B, polio, pneumococcal, no more rotavirus vaccine. We're going to go back to 70,000. If people listen to this, I hope they don't, we're going to go back to 70,000 cases of rotavirus diarrhea every year. 4 months, DTaP second, Hib second, polio second, PCV second, no more rotavirus. 6 months, no more Hep B, third shot. 12 months, no more Hep A, first and second shot. 11 years, no more second HPV. Meningococcal, no more first shot. 16 years, no more second shot of meningococcal vaccine. And then annual flu and COVID. Nope. No longer recommended for children. Why? What are the data that cause this? A comparison with other countries, which is irrelevant, which is not the way you do science and public health. Everyone should be outraged. You're outraged at the lady shot in Minneapolis. I'm outraged at that, and I'm outraged at this. You shouldn't be messing with it. And what is this going to do? People who don't know any better are going to say, oh, it's up to me. Like the flu and COVID is supposed to be shared clinical decision-making with your doctor. I don't do any stinking shared decision-making. I go to the drugstore and get my frickin' COVID and flu vaccines. And that's what everybody should do. Unless you're immunocompromised, then you have to talk to your doctor. I agree with that. So what's going to happen here is that a lot of people are going to say, oh, it's up to me. I don't have to get it. And it was never mandatory, right? But it was recommended. You went to your the doc who, if they were informed and looked at the CDC recommendations in the old days, they would say, okay, you're due for this. You should get this. You're the right age. You're the right group and all that stuff. Now, they're not going to bother. And what are we going to get? We're going to get more rotavirus because there's no more rotavirus immunization. We're going to get respiratory syncytial virus. It's really important to get these kids at birth, or at least give the mother a vaccine in the 3rd trimester so she can pass antibodies to the baby. No more. Hep B is going to be a ridiculous number of Hep B infections. Hep A as well, meningococcal disease. They've also eliminated the second dose of HPV vaccine. There was clinical study recently showing that one dose was sufficient, but I don't think it's definitive yet. So I think that's a bit too early. And there's no negative. There's no negative for any of these. There's no negative. There's only positives in prevention. So I don't know what to say, folks. Many states are saying we're not going to listen to these guidelines. We're going to do our own thing. And that's great. I hope the bloody red states get a brain of their own, get an independent mind, and do the right thing. I hope the doctors there don't use politics as an excuse for this. Every state should stick with the original recommendations. There's no reason to change. Do you think there's a reason to change? Tell me if you think so, but this is crazy. Okay. Let me go back to the comments and see if anyone has said, yeah, a lot of people have said things. For the bees, happy birthday, Vincent. Grateful for MicrobeTV and being here. Do we have a TWIV on bird flu basics, and are there precautions for cleaning bluebird boxes and purple martin gourds? We did do TWIVs on bird flu basics, and you can search. We did a recent one with Robert Webby from St. Jude's, and I think we covered a lot of basic stuff, but we've had a lot of others in the past. I think we even had a mini lecture here on office hours. Are there precautions for cleaning bluebird boxes? I don't think it's different for particular birds, but let's look it up. So the CDC, if you trust them, clean them regularly, wear disposable gloves, wash your hands with soap and water immediately afterwards, avoid unprotected exposures to sick or dead animals, and report sick birds or other animals to your state government. They say federal, but don't bother to report it to the federal because they probably won't do anything. That's right, MicrobeTV. Well, it's 16 years on YouTube, right? Yeah, because I started 2008 for 16 years. 16 years. It's also 16 years on YouTube. I must have started the YouTube channel at the same time I started TWIV. So MicrobeTV didn't come till later. I don't remember when that happened. But TWIV is certainly 16 years old. Kang writes, remember Thomas Hobbes said in The State of Nature, life is solitary, poor, nasty, brutish, and short. It's true. Humans have done well to live a long time. We're not evolved to do that. Patricia is in Massachusetts. Welcome. Plus, RFK Jr. is putting red meat back on the top of the food list. Yeah, to help the farmers, the cattle farmers, right? Yeah, at least he's also going after the real culprits of out-of-control metabolic disease in the U.S., sugar and processed foods. That's true. It's absolutely true. Yeah, there's the article on in the Times, if you have a subscription, where it shows the chart that I just showed you. Yeah, the facial expressions that they picked, crazy, right? It's one fetching portrait. Hello, Jessica. Yes, we haven't seen you in a while. Welcome. Happy New Year. Good to see you. Yep. Make America Denmark. Would be nice if we had public health. I mean, that's some people had that goal of having a universal health care, but it's very hard with the Republicans all against it because they don't believe that people should be given help. Look at the food stamps program and the supplements for Obamacare, all this stuff they're taking away. Cassidy Elizabeth writes, has all the power. He could impeach RFK at any moment. I don't think that he would get a majority. You need, what do you need for an impeachment? Just a simple majority? I don't know if every Democrat would go along. That's the thing. I'm not sure. I'm not sure. Soul Symphony, that scene from Monty Python, The Holy Grail. A vendor goes around carrying corpses on a carriage. The guy tries to get his living. Dad on should summarize how well we're going back in time. It's true. Yes, giving patients power. They've always had the power to refuse vaccines. The doctor always says to you, I think you should get this vaccine for this, because this. And then you could say no, or you could say okay. And I never said no, because I know better. Respectfully, why is the U.S. becoming anti-vax, pasteurization, healthcare? I mean, diphtheria, brucellosis, FMTV are bad things. Why are citizens and policymakers not on it? It's a complicated story, okay? Because in the U.S., a lot of people don't want to be told what to stick in their bodies, yet they will stick lots of things, including fentanyl, in their frickin' bodies and get addicted to it. This is why I have a problem with, addiction, fentanyl vaccines, because the same people will say, I'm not putting foreign substances into my body, and they've already done that. The vaccines are the best thing you can put into your body. Maybe second best after food, because you need that, right? So there's a bunch of people who are anti-vaccine for that reason. But the real reason is there are the leaders of the anti-vax movement who make a fortune selling crap that doesn't do anything and say vaccines hurt you. Buy my stuff instead. Robert Mercola, Andrew Wakefield, all these people make a ton of money. And RFK Jr. consults for law firms that sue vaccine manufacturers and makes a fortune. It's all about money. That's it in the end. And they take advantage of the fact that many parents don't know enough and they're scared. And they have this haughty attitude that nobody's going to tell me what to do. You know what? Tell me what to do, Doc. Tell me what to do, former CDC. I want to know what vaccines I should take, because I know the science says they're good. They help, they protect you against severe disease, and they don't hurt you. Yes. So, yeah, too much sugar, folks, which is really bad. And read Transformer by Nick Lane to find out why it's really bad. David, does Columbia offer a class on vaccinology? No, it's not. I have a class which has one lecture on vaccines, but that would be an interesting course, wouldn't it? So our province, which I guess is Canada, is moving to 1 dose for HPV vaccine. Yeah, there is some data, as I said, but there's no harm and having a second dose. And how do you know that 1 dose is good for everybody in every situation? We know 2 doses can eliminate 100% cervical cancer in many populations. Why would you mess with that with having one clinical trial? I don't get that at all. If it were nasty, sure, but it's not. It's harmless. It's not even infectious. It's insane. Merck has said you need 2 shots and they have 20 years of data. Right. And this other thing is 1 trial. Patricia, I think the whole Denmark thing is just a pit stop on the way to doing away with vaccines altogether. I think it was stated by Mark McCary. If you get away with vaccines, I'm going to come and wring your frickin' neck, okay? And they won't let me near them. I know that. There's no way you're getting rid of vaccines as long as I'm around. Hello, Barb Tries. Welcome. Durham 13C with the windows are open. Thank you for being here for us. I need you too. We all have to fight this crap. In a few years, when all these viruses take hold, Australia may have to list USA on the quarantine list like some poor African countries. Already, some countries are not allowing Americans to go there, right? Is that true? Or without a visa or something? Let's look it up. We have to see. Are countries preventing Americans travel. I can't find it. I just heard that some countries are restricting Americans to go there. Spamlet writes, I remember when every new academic year was heralded by reports of new students dying of meningitis. RFK Jr. is a murderer thousands of times over, and that's before the next pandemic hits. We have to hope that there's no pandemic while he's around, folks. Otherwise, it's going to be a disaster. Yes, they will rush to take any amount of supplement or vitamin Joe Rogan, Andrew Wakefield, Joe Mercola recommends. And yet something that's proven to work and save your life, they don't use. I think I read a comment on a podcast or office hours of someone older could not buy an HPV vaccine dose. Bummer. This is a hidden gem. Yes, if you're older, it's hard to do. You have to find a physician who is willing to do an off-label, and then you have to pay for it yourself, right? Yes, an HPV the vaccine prevents 90% of genital warts in men, as well as neck and throat cancers, which are horrible. Thank you, King C. King Pro. My birthday and MicrobeTV. RFK Jr. promised radical transparency. This announcement was made without discussion. Radical transparency. None of these guys have radical transparency. It's just a word that they think will impress people into believing that they're doing the right thing. If you believe that, I have a bridge to sell you. Oh, my God. Yeah, okay. HPV has been tested in a certain age range, up to 45. But a meta-analysis was just done. You got to listen to the TWIV episode, MN9159, okay? Because they found that in older people, it resolved HPV infection. And if you were going to have surgery, getting the vaccine before surgery improved your chances of resolving infection as well. So now it really pays to get the HPV vaccine at any age, not just up to 45 in the trial, because these other studies have shown there's a benefit even when you're infected. Thank you for donating. And anyway, we have a new series, Rasmussen Retorts. I hope you like it. We'll do another one next week. Provides a different perspective here on MicrobeTV. Now, impeachment requires 2/3 in the House, which we've done before, right? In the Senate. What is that? Oh, this is good. MAGA, manufacturing American-grown autocracy. Yeah. Okay, Nevitt says, correction, impeaching the U.S. president requires a simple majority in the House, followed by 2/3 vote of senators. Yeah, so that's a president. What about impeaching RFK? What is that? So Veronica says, why the U.S. is becoming anti-vax. Actually, all so-called first world countries have been doing this for some time now. I remember when France was the leader of the anti-vax movement, and whereas developing countries want to have vaccines. Yes, rugged individualism crap. You're absolutely right. And many Americans try to beat the system, think they can outsmart with another answer like ivermectin or hydroxychloroquine. Oh, it's cheap, and big pharma's trying to rip you off. Well, they are expensive, but ivermectin doesn't work, okay? Hello, Marco, coming in late, east end of Long Island, 2C. Maybe the history of vaccines as a course. That would be very interesting. I would love to teach a course like that. Actually, maybe I should just do an online course. Yeah, I don't have to worry about getting a classroom, blah, 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 having it approved at Columbia. I could just do a live course like we did a couple of years ago. Public health is in a crisis is all about mandates. Otherwise, it's only about personal health. Well, it's also from these people getting rich on being anti-vax, okay? Scoopax, hello from a microbiology student in Colombia. Love listening to your lectures while gaming. Keep it up. Hey, that's great. I never heard that before. Thanks for coming. Yeah, cervical cancer rates in Australia have plummeted since HPV on the schedule. This is a no-brainer, total no-brainer. Michael writes, here in Mexico, the former government claimed our health care would get as good as Denmark's, which didn't happen. We could increase our health expenditure as high as that of Costa Rica for starters. Okay, Sol, do your lab work. See you later. Yes, problem with old diseases is living memory has mostly left the population. The media gives so much information, so hyped and unreliable. Science is hard to understand, so few try. And Will says, Americans are blindfolded by personal freedom and ignore the reality of the social contract requiring regulations for the common good. Castle Hill, my lab is researching HPV. I was wondering if you had any good resources to help with that research. You know, you go to the TWIV where we talked about this meta-analysis, and that has a lot of stuff in it because it's very well referenced because it's a meta-analysis. So I would start there. You could also listen to my interview with Harold Zurhausen, right, who was the one who discovered that HPV was associated with cervical cancer. So I did an interview with him, but he has a great Nobel lecture as well, where he writes about the history of discovering it. Okay, let's do a mini lecture before we get too far into this here. So someone last time said, let me turn off that thing here. Someone last time said, can you give a lecture on your how you discovered the polio receptor? They said a podcast, but I'm going to do it here. And I'm going to compare it to how the norovirus receptor was identified. So this goes way back, okay? The poliovirus receptor is called CD155. It was identified in the late 1980s. Well, first, through a combination of virology, somatic cell genetics, and molecular cloning, okay? It's a beautiful example of receptor discovery before modern expression cloning shortcuts. So I was involved with this. This is what I did when I came to my lab in Colombia. And it was pretty old technology, although at the time we thought it was great. So it was cloned in 1989-90 with contributions from Akio Nomoto, Eckhard Wimmer, and me, our groups. Okay, that's the background. So first, a big clue was the species specificity of poliovirus. Poliovirus infects humans and higher primates, but not rodents. Mouse cells, though, are permissive to poliovirus infection. In other words, they have all the internal machinery to reproduce the virus. If you introduce poliovirus RNA into a mouse cell, poliovirus RNA is infectious. It will reproduce. But if you put virus on a mouse cell, it won't get in because they're not susceptible. So there's no receptor in mouse cells. So here's a paper by John Holland, 1959. I was 6 years old. Mammalian cell-virus relationship, poliovirus production by non-primate cells exposed to poliovirus ribonucleic acid. That showed that mouse cells, non-primate cells, were permissive. We already knew they weren't infectable. They weren't susceptible. So this suggested that a receptor was missing. Right. So that's the first clue. Second clue, people then made, and here are the authors who did this, human-mouse somatic cell hybrids were tested for poliovirus binding and infection. So what does that mean? You take human cells and you take mouse cells and you add a chemical that causes them to fuse. And so then what comes out are cells that are hybrids between human and mouse, and they can have different numbers of human chromosomes in a mouse cell background. Only hybrids that retain chromosome 19 support polio attachment and entry. So in other words, chromosome 19 has on it a gene, probably a gene for encoding the polio receptor. Okay, so that's nice, but we already knew that there was a gene somewhere in humans that could be produced in mouse cells. Okay, next. Monoclonal antibodies were raised against HeLa cells, and some of these block poliovirus attachment and entry into cells. And one antibody, it's called D171, recognized the glycoprotein present on susceptible cells but absent from resistant ones. So again, a susceptible cell has the receptor. That's the definition. A resistant cell has no receptor. So these antibodies so they're made against HeLa cells, and they're screened for the ability to block poliovirus infection of HeLa cells. And they do not recognize cells that don't have the receptor. So that's a good reagent, and we got hold of this. And this is what was needed to clone the receptor gene. So here's the antibody paper, 1985 now. Production of a monoclonal antibody against an epitope on HeLa cells that is the functional poliovirus binding site. So this paper describes the isolation and characterization of an antibody that reacts with the plasma membrane of HeLa cells. This antibody was selected for protecting HeLa cells against poliovirus infection. It extends to all three serotypes. It didn't block other virus infection. Didn't bind to cells from pigs or dogs or rodents, but bound to all lines of human or primate origin. And so 40 different clones, they have susceptibility of poliovirus correlates with binding of this antibody. So it looks like this antibody, as the title says, is against the functional poliovirus binding site. That's great. Then we published this paper from my lab. Kathy Mendelson, one of my earlier graduate students, and we did this in collaboration with Eckhard Wimmer. So this is a PNAS paper, 1986. So I'm at Columbia for 4 years. I go there in 1982, and I will go with the idea of cloning the poliovirus receptor gene. I had that idea. Well, it's not much of an idea. It's obvious, right? Everybody wants to identify viral receptor genes, but I wanted to do it in my lab. So here's the background. Mouse L cells, right? They don't have a receptor. What we did is we took those and we put into them human DNA. They call it transformed with HeLa cell DNA. And that makes the mouse cells sensitive to poliovirus. 1 in 50,000 transform. It's, in other words, 1 in 50,000 mouse cells that take up human DNA become susceptible to poliovirus infection. Then we use the monoclonal antibody directed against this poliovirus receptor site that I just told you. We chemically linked that antibody to red blood cells and then put it on top of the cell transformants and plates. And if the antibody bound to a receptor expressing mouse cell, you can look in the microscope and you can see the red blood cells bind. That's called the Rosette assay. And in fact, we could see binding of this antibody to mouse cell transformants. So then we cloned out a cell line called CM1 that was recognized by this monoclonal. Again, it's a mouse cell, so it's presumably got a human poliovirus receptor gene in it. And it was susceptible with infection with all three poliovirus serotypes, and infection could be blocked by the anti-poliovirus receptor antibody D171. Okay? So that's the basis for this paper. So now we need to clone the gene. How do we do that? We have these mouse cells that have the human gene in it. How do we get it out? So first of all, these are the numbers, the frequency of DNA-mediated transformation of mouse cells to polio sensitivity. In other words, we take human DNA, we insert it into mouse cells, and we do 3 different transformations here, each one 28 plates of cells. And you can see 1 of 28, 2 of 28, 2 of 28. Not very frequent, but it's there. In other words, positive means susceptible to poliovirus infection. And the polio-sensitive cells are 1 in 42,000, 1 in 60, and 1 in 65,000. So how do we get those polio-sensitive cells? Once you infect them, you kill them, so that's no good, right? So here's what we did. So we take a mouse cell that doesn't have the receptor. We put DNA from human cells. They now express the receptor, a few of them, like 1 in 50,000. So what we're going to do next to identify those cells is add our antibody coupled to red blood cells, our D171 that binds the receptor, and we're going to couple it to a red blood cell so that we can see the colonies on the plates and pick them up with a pipette. Visually see them and pick them up, okay? So here's what that looks like. So at the top, these are colonies of HeLa cells that do not produce, these are colonies of mouse cells that do not produce the receptor because we've added red blood cells and antibody, and the antibody is not sticking. But on the bottom, here's a colony that's producing the polio receptor. You can see all the tiny circles. Those are red blood cells that have been conjugated to the monoclonal antibody. You can see this under a microscope. You can you can scrape those cells off, and now you've purified a cell line that is a mouse cell line that contains a human poliovirus receptor gene. Very cool. So excited. I was somewhere in France, and my student Kathy called me, and she said, I got rosettes. I said, oh my God, that's great. Back in the day when you called people. I guess you still do call people. Okay, so we could pick up these cells. Now we have pure mouse cells with the human polio receptor gene in them. How do we get the gene out? Okay, so I have to tell you a couple of things. Now, this is an old picture that I made by hand in 1989 or something, right? So remember, we take HeLa cells that are polio susceptible. We take the DNA and we put it into mouse cells. Now, we don't just take human DNA and put it into mouse cells. We add also the thymidine kinase gene from herpes simplex virus, TK. That provides resistance to a drug called HAT, hypoxanthine aminopterin. Okay, this drug will kill cells unless the cells have taken up the TK gene. And when you mix 2 DNAs together, most of the time both DNAs get into every cell, not just one. It's called co-transformation. So we put the human DNA plus thymidine kinase DNA into mouse cells. And then we add the drug HAT. And most of the cells die, but you get HAT-resistant L cell clones. And so some of these are also going to have the polio receptor gene. So how do we find that out? We take this plate, we remove the cells with trypsin and make 2 plates. And we infect one with poliovirus, and we say, oh, 1 in 28 plates is positive. We take the SIB of that plate, which means the pair, and now we can clone out the polio-sensitive cells using those red blood cell assays that I just showed you right here. All right, we can. These are colonies growing in the presence of HAT, which is killing all the other cells that didn't pick up the TK gene. Okay, so how do we get the polio receptor gene out of here, out of these cells? Well, we use a trick back then, which was so cool. Nowadays, it doesn't matter anymore, but back then it was cool. There is a sequence in human cells, in human DNA, called an ALU repeat. It is the most frequent repeated sequence in human DNA. It's present about every 5,000 bases of DNA. In other words, it's likely to be next to any gene that you're interested in. And so we made a library from these cells. We digested the DNA, put it into a bacteriophage, plated it out, and then hybridized the plaques with an ALU probe, and that's how we pulled out the receptor gene. So let me show you how that works. You see how convoluted this is? So this is a southern blot. where we run a gel, an agarose gel. We take DNA of various cells. We digest them with restriction enzyme. We transfer the DNA to a nitrocellulose filter, and then we hybridize it with a radioactive probe against the ALU repeat. So you can see HeLa cells are full of ALU repeat, a big black smear, right? L cells don't have any ALU. It's a human sequence. And Kathy's CM1 cell line. These are the cells that received human DNA that she picked colonies that bound the red blood cells, bound to the anti-receptor antibody. These have far less human DNA, and still there's a lot there. But one of those bands must be the polio receptor gene. Now on the right is a growth curve showing poliovirus growth with time, PFU per dish. And one of these is wild type, and one of these is HeLa cells, and one of them is CM1. So the mouse cell transformant replicates poliovirus very well. All right, so this is still too much human DNA in these mouse cells to be able to efficiently clone it by making a phage library. So what do? Kathy takes DNA from CM1 and does another round of transfection into L cells. And she makes CM1.1.9, et cetera, et cetera. And so you go through the whole process. You make DNA from CM1, you put it into mouse cells with TK, you get colonies, you add the antibody receptor conjugate on, you pick colonies, and you grow them up. That's what each of these is, different colonies picked from those plates. And again, this is a southern blot. You can see all of the ALU DNA in CM1. And now, look, these cells have a lot less. They have a couple of bands of DNA, so no more than a few thousand kb of human DNA. So what we do is we take DNA from CM11, we digest it with a restriction enzyme, we clone it into a phage vector, we plaque this phage library on agar, on bacteria and agar, and then we transfer the plaques to a nitrocellulose filter. And then we hybridize the nitrocellulose with radioactive ALU DNA. And we pull out a DNA which, when put back into mouse cells, makes them susceptible to polio infection. Those cells bind the antibody D171. We have now cloned the poliovirus receptor gene. And I haven't thought about this in a long time. So it was very exciting for me to go back and remember all the things that we had done. So we identify in the coding sequence of that gene the poliovirus receptor, which is shown here, CD155. It turns out it's an immunoglobulin-like protein. It looks like an antibody molecule. And it turned out that a few other receptors that were being discovered at about the same time, HIV-1, the rhinovirus receptor, eventually adenovirus, they all are immunoglobulin-like proteins. So what do we do with this gene? Well, we made the protein, purified it, the poliovirus receptor, we mixed it with poliovirus, and we solved the three-dimensional structure of the receptor bound to the virus. That's shown on the right here. 60 copies of the receptor consistent with icosahedral symmetry. Another student of mine, Ribao Ren, took the gene that Kathy Mendelson had cloned, and he made transgenic mice expressing the poliovirus receptor. Now they are susceptible to paralysis. You can see this animal has a paralyzed hind leg after inoculation with poliovirus. So now we have a mouse model for studying poliovirus pathogenesis, which we did for many years, and it replaces the monkey for testing poliovirus vaccines, the live attenuated polio vaccine. So I feel this was my main humanitarian or animalitarian contribution that I stopped using monkeys to test polio vaccine. But of course, many people use this model to study poliovirus, and they still do today. Okay, so that took a while. Let me tell you the modern version of this now. If you want to identify a receptor of a virus, let's do norovirus, right? Because norovirus is a plus-strand RNA virus with an icosahedral capsid, just like poliovirus. There it is, and here's the paper 2016 now, many years later, discovery of a norovirus, a proteinaceous cellular receptor for a norovirus. What was the title of our paper? Let's see. Did I even put it here? No, I didn't put it here. Oh, that's too bad. So ours was cellular receptor for poliovirus, molecular cloning, sequence of cellular receptor for poliovirus, something like that. A new member of the immunoglobulin superfamily. I should have put it in. Anyway, so the same strategy, except we start with cells that are susceptible and permissive to norovirus, and we introduce the gene for Cas9 into them. Okay, I'm going to show you what that does. Cas9, of course, is the endonuclease that's used for the CRISPR-Cas9 system. So what we're going to do is do a genome-wide knockout of every gene in cells using single guide RNAs directed against every gene, because we've got the human genome sequence now, which we didn't have when I was working on the polio receptor. And we can say, let's knock out all the genes, one gene roughly per cell, and then let's find the ones that are resistant to norovirus infection, and then we'll see what gene has been disrupted. Okay, how do you do that? So you have a cell making Cas9. That's this protein behind the DNA here. So the cells are susceptible and permissive to norovirus. They're also making Cas9. And then you introduce a library of short guide RNAs. These include a sequence that will bind Cas9 and a sequence that will bind a gene. You put a whole library, tens of thousands of these into your cells. They're going to make double-stranded breaks. They're going to end up deleting sequences, and you're going to disrupt the gene. So then you simply take those cells, millions of them, and infect them with norovirus and see the colonies that survive. They're resistant to norovirus infection. You pick them and you ask what gene is disrupted. You can do that because the disruption, and you end up repairing the disruption with a sequence that you can identify using PCR. And so here's the result. So these are different genes that have been knocked out in cells, and the blue color and the blue color are cells that are still susceptible to norovirus infection. The red are now resistant. So you have a whole gradient of cells, but you have one at the top here, CD300LF. That's the name of a gene, which when disrupted makes the cells resistant to norovirus infection. And you can see that here in this growth curve. So wild-type norovirus, here's PFU per mil with hours. You can see they're growing. The knockout of CD300LF, now the virus is not replicating. All right. And they do this with two different norovirus strains. Here on the left, they put viral RNA into these cells to make sure no other defect has happened. So this is wild-type cells. They reproduce norovirus well. This is the deletion of CD300. They still replicate norovirus, and then you return the gene to those cells, and it still replicates quite well. They have a monoclonal antibody against cells that blocks norovirus infection, and these knockout cells do not. These are adding soluble protein, receptor protein to infected cells. And so you can see adding increasing amounts of the receptor protects the cells. You have viability 100% here. So now we're going from 0 to 100% with two different strains of norovirus. So in other words, the protein can bind virus and block infection. And finally, they knock out the gene in mice using CRISPR-Cas9. And we have the knockout of this CD300LF. These animals are fine, no norovirus in the feces, and the wild type mice, which are plus for that gene, they make virus and shed it, and also the heterozygotes are still susceptible because they have one copy of the CD300 gene. So all of this shows that this CD300 encodes a protein that's a norovirus receptor, and I didn't have a picture for you, but it's an immunoglobulin-like protein, very much like the polio receptor. But you can see it's much more straightforward now because we have gene editing and we have the whole genome sequence, so we can easily identify virus receptors. Why do we care? Well, for poliovirus, it explained species tropism. It let us make transgenic mouse models of polio, and it gave insight. Subsequently, we studied poliovirus entry and how the receptor participated in it, and this provided insight into the coronavirus entry. So that is how we identified the poliovirus receptor. Very exciting. Okay, let's see. How close are we to bird flu mutating? Well, why do you think it's going to have a 35% mortality rate? Okay, so it could sustain mutations that make it go human to human. We have no idea when, how close we are. But you're making an assumption that it will have a 35% mortality. And that's just wrong. There's no reason to assume it's going to be the same. So, yes, there is an mRNA vaccine ready, but as you know, RFK Jr. does not like mRNA vaccines. He wants to make formalin inactivated vaccines, which are 100 years old. So let's see what happens. This is going to be a tragedy, I think. Hello, Joel. Welcome. Do a live course in charge. No, it'll be free. Always. Abdur says, we kind of do during med school a mix of biochem, physiology, and micro is where we learn about vaccines. Yeah, but there's not a dedicated course in medical school for vaccines. No. Wasn't there some podcast that did a series of on vaccines? I don't know. I love listening to your podcast. Thank you. I appreciate it. Oh, Mark wants to know about H. pylori and oncogenesis. I think I've got that written down also. But we'll do that on TWIM. All right. Marco likes the idea of a course on the history of vaccines based on the discussion you had a while back about polio when there was no indoor plumbing. It was eye-opening. Hello, Sandy. Welcome. How did they find a chemical that made human and mouse cells fuse? Oh, it's been known for years that you add this to cells and the membranes fuse. Let's see. I'll tell you the name of it. It's called a cell fusogen. And the one they used, they also use it for making monoclonals these days. They fuse cells. Polyethylene glycol does it. Yes, R.I.P. Akio Nomoto, who was also, he was our competitor on that. He was working independently and did it about the same time. Yeah, I was the technical expert to the author of The Immortal Life. Yes, you'll see I'm mentioned a few times. What biotech would you wish you have today, if anything is possible? Well, one thing I think would be great is to be able to predict the structure of a protein from the sequence, but get it right, always right. Not, I like AlphaFold 2 is great, but it's not always right to get it right completely. What tech? I don't know because I don't have it. And right now, you can do almost anything you want. It's amazing. Yep. This is honestly beautiful across the board. A red blood cell assay, the transfection, the whole discovery. Yeah, it was cool. We had a good time. What's my old student, Kathy? She is a full professor at Columbia Medical School working on kidneys. Yeah, working on kidney development, actually cancer. She gets a lot of money for that. She's great. She's doing great. How would the isolation and cloning be done today? You would take HeLa cells and do a CRISPR knockout library like they did for norovirus. Isolate resistant cells and identify the gene that way. It'd be a lot faster. Do you worry that losing these intermediate techniques, most people are becoming more consumers? I do. I think, there's a lot of ease that you get, ease of performance, but I think if you're the people who end up making seminal discoveries, do something different. Yeah. Yes, we made the primate-specific virus infect rodents. So the mouse, the transgenic mouse that we made, is a gain of function for the mouse. Okay? For people out there who don't understand what gain of function is, it's giving a new property to anything, any living organism, a virus or a living cell or an animal. And we gave the mouse a new property, so it was a gain of function. And nobody cared because they don't know anything. They don't get it. They don't understand. And there was a possibility that that mouse could get out and maybe establish a reservoir. But it turned out it wasn't orally susceptible, so it wasn't an issue. So, it's amazing that Kathy could manipulate blind, right? She totally did it blind, using an antibody bound to red blood cells and then using this ALO sequence. It was amazing. How can one learn about investigating intentional exposure events, and how does one come up with a cure? We're doing that in Uganda, right? Isn't that what RFK is? He's testing a Hep B vaccine, and it's not intentional exposure, but he's got a placebo group that's not getting vaccinated, which is unethical because we know the vaccine will prevent Hep B. You cannot deny that to a control group, and yet he's doing it. Tuskegee. So look up Tuskegee, right? Where they exposed black men to syphilis. And that's totally unethical. And there are many examples throughout history. For syphilis, you have an antibiotic, fortunately, but that's a very sordid history there. So just look it up. You'll find it. Yep, science is incredible. I love it. But you know what else is incredible? Poetry. And here on Office Hours, we have developed a love for poetry. At least I have developed a love for reading it to you. You guys seem to like it. So we are going to read a poem. So actually a couple, because I really like E. Cummings. My mother used to teach it. She was an English teacher, and she would read it in her class and go over it. So he broke from traditional poetic forms. He experimented with syntax, with punctuation, with visual presentation. He only used lowercase letters. He had unusual line breaks, creative spacing to emphasize words, disrupt traditional grammatical structure. So things may sound a little weird, but that's part of the effect. So these are pretty short, so I'm going to read a couple. And I want to show you some of the ways these words are laid out on a page. Now, maybe there's not a lot of E. Cummings fans there, but listen, just sit back and relax, and you'll like it. So a couple of poems. The first one, a very famous poem of his. I carry your heart with me. I carry your heart with me. I carry it in my heart. I'm never without it. Anywhere I go, my dear, and whatever is done by only me is your doing, my darling. I fear no fate, for you are my fate, my sweet. I want no world, for beautiful you are, my world, my true. And it's you are whatever a moon has always meant, and whatever a sun will always sing is you. Here is the deepest secret nobody knows. Here is the root of the root, and the bud of the bud, and the sky of the sky of a tree called life, which grows higher than soul can hope or mind can hide. And this is the wonder that's keeping the stars apart. I carry your heart. I carry it in my heart. So you notice the syntax is a little funky, right? But let me show you. Let me see if I can make this bigger. Well, I can't. Anyway, I want to show you. So this is pretty standard verses, but look here. I fear is on a line by itself, and it's all the way indented, and it makes you focus on that word, right? Because that's really important. I fear, and then he says no fate. Okay. Another one. Maggie and Millie and Molly and May. This is fun. Maggie and Millie and Molly and May went down to the beach to play one day, and Maggie discovered a shell that sang so sweetly she couldn't remember her troubles, and Millie befriended a stranded star whose rays 5 languid fingers were, and Molly was chased by a horrible thing which raced sideways while blowing bubbles, and May came home with a smooth round stone as small as a world and as large as a loan. For whatever we lose like a you or a me, it's always ourselves we find in the sea. This is, in terms of lines, it's pretty straightforward, all lowercase again. But, for example, whose rays 5 languid fingers were. Instead of saying whose rays were 5 languid fingers, he puts the were at the end. It totally freaks you out, but it really emphasizes, right? That's why he's so cool. Okay, another one, and this one I'll have to show you the structure when I'm finished with it. It's called I Have Found What You Are Like. I have found what you are like, the rain, who feathers frightened fields with the superior dust of sleep, wields easily the pale club of the wind, and swirled justly souls of flower strike the air in utterable coolness, deeds of green thrilling light with thinned new fragile yellows, lurch and press in the woods which stutter and sing, and the coolness of your smile is stirring of birds between my arms, but I should rather than anything have, almost when hugeness will shut quietly, almost your kiss. So look at this. I don't know if you can see it again, but this is really unusual. Let me see if I can make it bigger. Okay, so we have really unusual structure here, right? We have a line and then it skips, and then we have an indent, which is weird, right? But then down here, deeds of green thrilling light with thinned is a separate line. New fragile yellows lurch and press in the woods which stutter and sing. This little bit here. He breaks each word onto its own line. Now this is common nowadays in poetry, but when he was doing this, nobody did it. He was totally unique. That's why he's so cool. Okay, one more. This one is, may I feel, said he. May I feel, said he. I'll squeal, said she. Just once, said he. It's fun, said she. May I touch, said he. How much, said she. A lot, said he. Why not, said she. Let's go, said he. Not too far, said she. What's too far, said he. Where you are, said she. May I stay, said he. Which way, said she. Like this, said he. If you kiss, said she. May I move, said he. Is it love? Said she. If you're willing, said he. But you're killing, said she. But it's life, said he. But your wife, said she. Now, said he. Ow, said she. Tip-top, said he. Don't stop, said she. Oh no, said he. Go slow, said she. Come, said he. Said she. You're divine, said he. You are mine, said she. A lot of parentheses in this to give it some kind of emphasis. But, of course, you see he writes a lot about love. He was very into that. But this one, because he's obviously having an affair with this lady, and she says, but your wife, said she. So this is a reflection of his own life, because he had an affair with a woman who was married, and the guy she was married to knew about this affair. He was okay with it. And in fact, they even had a kid. And eventually she got divorced and married E. Cummings. And then she went to Europe on a boat and met another guy and fell in love with him and divorced Cummings and married him. So he's used to non-conventional arrangements, I guess. Anyway, I really like E. Cummings. It takes a little bit of getting used to, but if you look at it as you're reading it and see how the words are jumping around, you really like it. So my mother taught this in high school, and she would tell me about it. And she said, we're going to do E. Cummings today, and I just thought it was so unusual. So you should check it out. It's really cool. What's my mother's name? She was Christine. Keep reading us poetry. Oh, I read this at my friend's wedding. Which one? I bet it's the I Carry Your Heart With Me, right? It's probably. Okay, in Tuskegee, they did not infect men with syphilis. Rather, they studied men with and did not offer them antibiotics. Okay, thank you for the correction. Yeah, the Nazis did a lot of crazy experiments on innocent people, yeah. I'm glad you like my poetry choices. Jabberwocky. I could read Jabberwocky, yeah. Yes, Archie the Cockroach, the fictional character created by poet Don Marquis, was also unable to use the shift key on a typewriter. Right, all lowercase like this. It's really cool. Seeing the structure of an E. Cummings poem makes me excited. Thank you. You're welcome. The structure is really a big part of it, and I don't know if you could see it, but you should go. They're all online. You can find these and take a look at them, and they're really fun. Yes, also the run together words, right. I like this one in the Maggie and Millie and Molly and May. It's a fun one, right? May came home with a smooth round stone as small as a world and as large as alone. Isn't that great? As large as alone. Having an affair is one thing. Cheating is next level. It's an artist, I guess. I am not familiar with Wordsworth Ode on Intimation. So we'll get to Wordsworth probably at some point also. Archie and Mehitabel by Don Marquis. Yep, right. For the Bees, Where's My Vax Gratitude shirt? Love the poetry. I would very much like a history course on vaccines, maybe one oriented for non-scientists and a second course for scientists. Well, I don't know if I can do both. I can start one. I can try and make it. Look, I think our podcasts, at least sometimes, get both audiences. I think this stream can get both, because here we have some scientists and we have non-scientists. So I think I could do it in a way to get everybody interested. Because you know what I find is that once you're out of your field as a scientist, everything is learning, right? So I do This Week in Neuroscience. I don't know anything about neuroscience. I can learn from my podcast. So we'll see. Yeah, office hours just got sexy, right? That poem. Yeah, what was it called? May I feel, said he. It is so funny, that poem, right? May I feel, said he. I'll squeal, said she. It's just delightful. Yes. From stem to steam. That's great, John. That is great. Visto writes, are there any poems with a mix of double-stranded and single-stranded stanzas with overlapping reading frames? Always so funny, man. That would be great. You know, there are people who set DNA. They play music with DNA, right? They give each base a different note, and then they play different sequences, and sometimes they're interesting, especially if you have repeat sequences, right? But I'm not aware of this, but it's really funny. I think it's really good. I shouldn't have my speaker on with this one while still working. Well, it's over, and it was only a little bit. Yeah, maybe Visto could write that poem himself. That would be funny. E. Cummings. Any other questions, folks? I'm done. But I'm happy to continue for another 10 minutes. Okay. Well, there's a 30-second lag. Let me wait. Yes, that was pretty brilliant. From stem to steam. Why 4 bases? Why not 3 or 5? Is it known that 3 or 5 were tried and evolved away to 4? We don't know anything about the early evolution of the genetic code, right? I think a lot of people have looked at this, right? So it probably evolved before the evolution of protein synthesis. That's one of the ideas, right? And so it may have been that it made the correct for RNA-based replicators. Remember, early in evolution, there were just RNA molecules that could replicate themselves. They weren't translated into protein. And so maybe 4 bases can be copied with high reliability, whereas 3 or 5 would not be. And so that was possibly the selection. Excuse me. But there's some papers that examine this, right? But the key is that it happened before protein synthesis. So that is something you have to take into account. It's a vaccine against athlete's foot. It's a fungus, I'm sure. Excuse me, I got a tickle in my nose. I used to know the name of the fungus. I'm going to look it up. Tinea pedis. I don't see why not. I don't see why not. Hello, Jill. Welcome. Read it, play back and listen to the poetry, if anything. John writes, if anyone needs something pretty to see, there's photos of a candlelight vigil going on at the scene where it happened in Minneapolis today. A lot of people are there. Lori loves the new show with Angie. That's great. I hope evolution eliminates human stupidity one day. I don't know. It hasn't so far. Yeah, and why 21 amino acids, right? What came first, proteins or DNA, RNA? RNA was first, that's for sure. And then probably protein synthesis, and then DNA. Frankie says, I've seen a paper on why 4 bases. It seems these 4 have a tendency to stay uncharged at the relevant pH. Charged bases would make pairing quite difficult. All right, folks, that's it for another office hours. Let me thank my moderators. We had Les here. We had Vanity Nutrition. We had Tom Steinberg. Thank you so much for moderating tonight. And thanks all of you for being here. I don't know if you haven't hit the like button, please do so before you go. We'll be back next week. What's the date on next week? Yeah, it's the 14th of January, 8 p.m. Eastern. I'll find another mini lecture. I'll find some more poems. I'll find some more news to rant and rave about as usual, and I'll enjoy all of your company. So please, until we see you next time, be safe. Thank you and good night.